Well, it's really a distinct pleasure for me to be here. I was really tickled to hear the award ceremony because it reminded me of the Oscars. But instead of thanking parents or mother and father and the producer, I heard an emotional acknowledgement of a parental chip for your system, and a touching embrace of the number of terabytes that it really serves. So it was a whole new world for me, to say the least. Before I start on the science part, and I'm going to be presenting to you the science of my colleagues at the Jackson Laboratory, I realized when I started the job, despite the fact that in the genetics world, the Jackson Labs is extraordinarily well known, it's an institute that is not well known to the general public. So I want to introduce you to the Jackson Labs. It began in 1929 with a few very intrepid pioneers who believed that genetics can be studied in a mammalian system. Now, you think this is a really bizarre statement, but genetics as a term, as a science, really began in the turn of the century. Before the turn, in the 1800s, there was no term called genetics. And it was also thought by horticulturalists and animal husbandry people that inbreeding a mammal could never occur because in their practice it would always lead to a very unfavorable strain. Well, our C.C. Little, who began this, was one of the pioneers in genetics, but using the mouse as the model system. He decided, for many reasons, which I'll tell you maybe in our private session, to go to this small town in the far east corner of the United States, Bar Harbor, with a population at that time less than the 5,000 that you see here today, to start this research institute. But going forward, we're now not your grandfather's Jackson Laboratory. We have 1,900 individuals in 3 different campuses. Our major one is in Bar Harbor, which is focused on mammalian genetics. We have a new campus in Farmington, Connecticut that focuses on human genetics and genomics, and a production facility to provide the 3.5 million mice to 22,000 laboratories in 62 countries in the world to pursue their research. We have a bipartite mission. The first is really our commitment to our own research, which is to discover precise genomic solutions for disease. And then the second, which is what we're really quite well known for, is to empower the global biomedical community, including many of you in the audience, in your quest to conquer human disease. Bar Harbor, our laboratory abuts the beautiful Acadia National Park, and it's a wonderful place to do, and a contemplative place to do some fundamental science. In 2014, we opened up a 200,000 square foot building in a 17-acre campus right adjacent to the University of Connecticut hospital system that is focused on human genetics, genomics, and computational biology. In this little town of 5,000 individuals, we built our own Nobel Prize in George Snell, who in 1980, because of the experiments using the genetics of the mouse, uncovered what is the fundamental of all transplantation biology. And he received a Nobel Prize in 1980. We have now 69 professors. We have major NIH-designated centers in cancer, in aging, precision genetics, in knockout mouse, one of the 3 in the world, Alzheimer's disease, and the ENCODE, which looks at the complexity, which is the title of my talk, complexity of the genome in terms of the higher order organization. At the Jackson Laboratory, because of our commitment to 2 model systems, the mouse and the human, we really do believe mouse genetics is the other half of human biology. Our focus is strictly on complex genetics and functional genomics towards the understanding of the human condition, like aging, and to cure human disease. We have no specific disease we're focused on solely, but we're opportunistic to take our technologies, our platforms, to attack the disorder that we can most quickly find cures for. Now, let me define my concept of complexity here. All of you are masters of complexity because of exactly what you do. Just as an image presentation, you see that these are blocks of color. When you piece them together, they're pixels in a color combination. And what you see then is actually progressively, with higher complexity, greater recognition of form. You know this already. In genomics, we have ATCG, which is your nucleotide. You sequence them together in 3 billion base pairs, which is your genome, into a string, a linear string of information. But that information is actually organized in a highly complex, multi-dimensional space in how the proteins interact with each other, and even how the DNA fragments interact with each other. You know, the paradox is that with our technologies in genomics, we can know every single genetic detail, every single nucleotide, and measure any phenotypic output with many of our technologies. But the challenge now is really, truly, to how to measure, simply how to measure complexity, to understand the complexity, to have a nosology about what is a complex system that is different from the other, how to create complexity so we can prove a principle, and how to master that complexity in what I won't be able to talk to you about is synthetic biology, which I believe is one of the brave new futures of biology. The problem with mouse models is not the mouse itself. It's really the problem of how mouse models used to be used, which is really like you all know that if you had to deconvolute your computers into adding machines, it would be crazy. Nobody would ever think of that as a solution to high compute problems. So you have seen a lot of criticism about mouse models, but this is the problem. The biologists have used one inbred line with a single mutation that is discovered in one patient to represent an entire human disease, like Alzheimer's, like diabetes, like ALS, like Parkinson's disease. I would say it's akin to taking one genetically ill individual with a private mutation in cystic fibrosis that causes cystic fibrosis, and then try to generate an entire drug panel for diabetes using that one person. It makes no sense whatsoever. But that's how the biology community, for no nefarious reasons, have been using the mouse models. You fast forward today, and we have unique and remarkable technologies, but the scale that is necessary requires the kind of infrastructure that only the Jackson Lab has. 1,300 individuals that are focused nothing but the welfare of our mice and also the experimentation of mice. With 1 million square feet of space, with 1.5 million mice living at any one time, which, by the way, is more than the entire population of the state of Maine. Now, I'm going to give you an example from Greg Cox, where he, using a mouse model, identified a mutation, a neuromuscular disorder causing neuromuscular degeneration, as you can see in the hind limbs of these mice. And by mapping and by sequencing, he found that it was the mutation was in the immunoglobulin mu-binding protein 2. And the name itself is somewhat irrelevant. But what was found, he found this first in a mouse, but because of the publication that he had that showed a phenotype that resembled a single family's phenotype found in Europe, the European colleagues called him up and said, could this be the same? And so when they worked it out, that disease called spinal muscular atrophy with respiratory distress, which affects kids with muscle wasting, but they waste in the center instead of from the periphery first. And many of them die of respiratory failure, okay? And it turns out it was exactly the same gene. Now, in order to identify the details of the genes, the biology of these genes, he crossed the mutation in this one inbred line with another inbred line. This is a standard mapping procedure that you have of mixing the genetic material to map the mutation. And what he found very peculiarly, this is actually what you see here, is a single litter that supposedly should have the disease in all the all the pups. But instead, you see that some are severely affected and others are not. In fact, what he did was to find out was that the genetics, the background genetics of that other strain completely blocked the negative effects of the mutation of this IGH-MBP2 mutation. And when you take, the picture that you see here is a section of the sciatic nerve itself. This is the nerve going down to the leg. And each one of them is an axon going down. In a healthy animal, you see that there are a full complement of numbers and size of the axons going down. With a SMART, they already are degenerating because it's a neuromuscular disorder, and actually it's a motor neuron disease, so that the origins of that axon is actually degenerating. You see it's shrunk significantly. But when it's crossed with this other strain, it completely normalized the phenotype. That is, it cured the disease by just this mating. Now, the big question for us as geneticists is, what's the gene? And by using this kind of genetics, which you can never do in the human population, we narrowed it down to chromosome 11, to chromosome 13. That is the rescue gene. And it found to a 400 kilobase region, which is now narrowed down to about 20 kilobases, and we have 3 candidate genes. Each one of them with very unique characteristics that make it, would have never have been able to figure it out by just sequence alone. So you need this kind of approach. Now, how do we apply this into the human condition? Well, this is work by Nadia Rosenthal, who is the scientific director in our Bar Harbor campus, and she's working on the genetics of cardiac regeneration. Now you know, we know in human populations that heart attacks are unfortunately common. But when you do get a heart attack, there's different outcomes depending on the amount of stress that you've had before, your gender. Male, female outcomes are very different, and also your lifestyle. If you're a smoker, you have a worse outcome than if you're a non-smoker. All these things are really very, very important in defining the variety of the outcome, not to speak of the genetics alone. So what Nadia did was ask, are there variances to the outcome of the myocardial infarction, which with the endpoint is dead tissue and heart failure, that is based on genetics, by simply looking at the outcomes in different strains of mice. Now, differences in cardiac repair, which we can actually look in a highly refined manner, can they tell us about the genetics of the diverse outcomes in humans? Now, how do we do this? Is instead of using inbred mice, that one single genetic background, that because every sister and brother of that inbred animal is identical because they're identical twins. But we redefined genetic diversity in the mouse by taking 8 founder mice and allowed random crosses to take place. These crosses will generate then variances like you have in the human population, and in fact, more variances than you have in the human population, with 45 million single nucleotide polymorphisms, more than in the human population itself, with 2 million indels, which is structural abnormalities. But the most important part of this, because this was done with 8 founder mice, every minor allele frequency is 1/8. That gives you tremendous power in terms of the mapping capabilities. And this is just, each color represents a strain origin of a chromosome, and you can see that we can actually generate immense diversity. Now, the challenge is that in terms of phenotyping is that mouse hearts are much smaller. They beat 400 times per second. And were thought to be resistant to heart failure because the experiments were done on one inbred strain alone. Well, what Nadia showed is by using this diversity set that in one substrain you have survival of 95%, and another you have 60% survival. One you have no dilatation and robust recovery of the heart, and the other, even if you recover, even if the subject recovers, the heart is dilated, there's weight loss and subsequent heart failure. Well, there's variability in terms of the scar, in the vascularity, and in the immune system. And when she scanned across numbers of different combinations, genetic combinations, those combinations gave a gradation of severity in a reproducible manner. Now, because I told you we can do some phenomenal mapping with this, she did the mapping and found that the mapping of heart failure is with this model system was on chromosome 5. When she did the fine mapping, she found a very small region that could be identified, associated, and there was one candidate gene that was really remarkable. In her practice eye, she zeroed in on this myo18B. Now, myo18B is localized to the actin stress fibers that populate muscles, both cardiac and skeletal muscle. But its embryonic expression is localized to the heart. Now, that's the immediate first indication there's some importance to this. But then what happens is that a knockout of this myo18B in the past was shown to induce heart failure, and the animals never survived to birth. So it's embryonic lethal, okay? But what was found was that in a clinical case, one single clinical case, a premature stop codon, which crippled the myo18B gene, was in a family associated with cardiomyopathy, which is similar and inducing heart failure. Now, the normal variations, that it's not the total knockout, but the normal variations of the myo18B appears to make an animal more susceptible to heart failure after myocardial infarction. This information is now being moved into the human setting, asking the question, using powerful sequencing approaches, whether or not the myo18B in humans is also associated with the variability of outcome. Now, that I showed you very early the problems of mouse model 1.0. We're moving into mouse models 2.0 because of these diversity sets that we've generated over the last 10 years, and also because of the new technologies you may have heard of, which is CRISPR-Cas technology, which allows us to have actually precise changes in the genome that is highly targeted without going in different parts of the genome. So also with the masterful work in the clinical sector of sequencing individuals with serious disorders, we find that we can actually identify all these mutations that might be candidate mutations in candidate genes in humans, which, by the way, we're finding hundreds to thousands of these in any one specific disease. How do you then, by association alone, by statistical association, make any claims? Because number one, they can be rare, and number two, there's so many of those. So one has to then move into a model system. And so what we're doing now is producing mutations in different genetic backgrounds in a mouse, phenotype them in a highly rigorous manner, giving us an understanding of, like you saw there, cellular mechanisms, ability to extract the cells to do actually the cell biology, and then use these cells to do drug screening. We bring it back to the clinical sector, and then in this iterative manner, find some ground truths about cure. Now, when we look at the genetic variability, as I showed you, not only does it allow you to map with precision some of the effectors of that alter the outcome of a mutation somewhere else, but you also can actually identify potentially targets for therapeutics. Instead of attacking a specific mutation, which may be not possible to do, you can actually activate a modifier gene, so it's an orthogonal therapeutic intention. So that's what we're doing quite a bit. Now, here's another problem. Complexity, where there's one disease and many genes operating all at the same time. How do you model this when the technology can only have you model one gene at a time in one strain at a time? Now, this is from the web, and it shows the genes that have been reproducibly involved in type 2 diabetes. There are about 75 to 90 of these, depending on the intensity of the association. Well, we at the Jackson Labs, and this is work by Albert Cheng, is creating new sets of technologies that we can multiplex the different mutations, changing expression levels both up and down in the mouse simultaneously, which had been in the past not possible. So one of the first things we did was to show that instead of doing the tedious microinjections, we can actually electroporate embryos with these guide RNAs. What that does is it frees us up from the artisanal approach of single ova injections to one that we can do in 96-well format. The second is the work by Albert, where we're using a hybrid system in the CRISPR-Cas process to allow for multiplexing. And how does he go about doing it? But you know, Cas9 is really the engine for the mutational generation. The guide RNA actually guides the RNA species and the Cas the Cas moiety to the specific area, and then something happens. Usually a break takes place, and the repair mechanism of the cell then either repairs it with a mutation or repairs it with an insertion. Well, what Albert did was actually did one step further, and in the tail of the RNA put a series of signature motifs, each one representing our recognition sites of these, what is an RNA binding repeat element, there's protein repeat element called the puff elements that actually recognize specific motifs. Now you can imagine this becomes a digital code for stacking together different motifs, different protein motifs, repetitive motifs, to recognize different nucleotide motifs. So you have a target, and you have a cargo that can be loaded in a highly specific manner. So you can multiplex. In other words, in one cell, you can have 3 different orientations simultaneously without doing serial transfection. And you can multimerize 1 copy to 1,000 copies if one were the case, or put complex formation. So you can put a DNA repair element, and you can put an element that would open up the chromatin simultaneously. So we can do this in genetic backgrounds simultaneously to activate and repress, to actually mark genes with labeling, or to delete genes as well. Now, I'll just give you one example that it does work using 2 different targets, which is OCT4 and SOX2, simultaneously in the cell of a mouse. And then depending on the familial tags that are in back, he could actually switch up, as you can see in the red, the OCT4, or switch down, is seen in green, the SOX2, and you can dial them up and down. So what is the combinatorial limit to this? It's 60,000. So you can imagine the theoretical limit of this technology is to change the genome in subtle or dramatic ways, 60,000 loci at a time. Now, we're far from there at this point. We're still at the adding machine portion, but we hope that we'll be able to do high computers with this kind of approach. Now, the idea here is that instead of having a mouse for every mutation, we actually can do a mouse for every abnormal network. And that will raise the complexity, of course, of our experimental systems, but it gives us higher resolution in terms of the disease that we have. Now, I'm going to give you another example where your technologies are absolutely essential for how we are going to conduct business in complex neurophenotypes. Of course, you know about the whole issue about neural networks. But this is work by Vivek Kumar. And it's about a complex behavior. And we took the behavior of mouse grooming, how actually mice groom themselves in a stereotype manner. And when there are neurologic defects, they have this stereotype actually go off. Now, if you see this here, is that this is the standard cleaning grooming behavior of the mouse, okay? If you do it in slow motion, you can actually see the components. One is this rotation, elliptical strokes. The other is the unilateral strokes going down. The next is flank licking. I do that periodically at night. Really, I don't. So you can see in real time that that's how it works. What he was able to do is actually use, in a cage, optical sensors, and then deconvolute this using neural nets, okay, elliptical strokes. And I want to show you that this is actually a mouse that has a subtle neurologic abnormality. In fact, it's not a neurologic, it's a behavioral abnormality. And you notice that the behavior is actually different. Well, let's talk about the neural net, the physical networks of grooming behavior. It's complex. It's not one locus, it's a bunch of loci. And in human disease, you have on the x-axis a difference in locomotor activity. You know darn well, Alzheimer's and Parkinson's, they move very slowly, low motor activity. But if you have frenetic individuals who are psychotic and who has Tourette's syndrome, they have high activity. Now, the problem is that in terms of how they're patterned, the complexity of their patterning, okay, that y-axis is not captured in any way with the current tools that we have, except by a clinician saying, this is a classical Parkinson's pill rolling and shuffling gait approach, okay? So what he did was to convert this into grooming classifiers using neural net approaches and was able to classify this complex functionality into something visual and reproducible. And in that manner, in the different strains, recall, I told you about the strain differences. He can show that reproducibly, different strains have different grooming behaviors, and that by using genetics, we can actually deconvolute the genetic components of this grooming behavior. This grooming survey across a whole number of the strains that we have 9,000 different strains at the Jackson Labs that are stored. Now move this into the clinical world, and this is work by Peter Robinson, who just joined us from Berlin. And he's been working on the big data problem in the clinical phenomics, which means you have all these abnormalities that you see here. They're visual abnormalities. In pediatric genetics, it's called dysmorphology. They have people who are called dysmorphologists who look at children and say, aha, this is the syndrome, okay? How do you then make that into a quantitative scale so that you can compute against the large databases that are there? And so Peter has pioneered methods to encode the meaning of clinical data, which is not trivial. Because, as you know, the ICD codes that you work on, they're a billing code. They're not scientifically structured. And so what he did was actually to deconvolute human physiology and physiognomy into compute possible approaches. But of course, as you well know, how do you then define a syndrome when there's such overlap and so forth? So these algorithms have been constructed. They are just in the first prototype phase, and we hope to make that more important. But he already has applied it in several cases. This was published in Science Translational Medicine, where 40 individuals that had no diagnosis after extensive clinical genetic evaluation. Okay, we see that actually in clinical medicine quite a bit. And in those individuals, they, using these algorithms, were able to derive definitive diagnosis in about a third of them, using, looking at databases across species, especially in the mouse. And for instance, this individual, 17-year-old male with this, again, very complex phenotype, and including brain phenotype, skin, reflex, even ocular phenotype. It was flagged because there was a correlation, though not a one-to-one association, with a mutation in a mouse, in several mouse strains. It is actually the cloud of phenotypes that then converge on this one mutation. It turns out to be a CYP27A1. Now, you could say, well, why couldn't you just sequence the genome? Well, they did. But there's so many abnormalities compared to the reference genome, which one are you going to choose when you have no clue as to the origins of this disorder? So the important part of this is because of the knowledge of the biochemistry, they were able to start actually a nutraceutical approach, and the individual was improving. So now we're going to expand that because the Jackson Laboratory is the home to the mouse genome informatics. This is a singular place where all the mouse phenotype and genotype information are housed. We've been doing this for the last 25 plus years. There's 65 individuals dedicated to doing this. But we want to mine this information in such a way that they're now going to be useful for the discovery of causal mutations in a human setting. My final slide is really to show what I consider is a challenge. At dinner last night, I mentioned I spent a short sabbatical at the NCSA, which is the National Supercomputing Center in Illinois. And they taught me a lot, and I was there to teach them about genomics. That was about 10 years ago. Every question, they said, okay, hit me with things that we can do. Every time I said, well, you could do this, they said, well, that's not a big enough problem. It got to a point where there was nothing I could tell them was a big enough problem. And maybe that's where you are seeing this. But where I think it is a big problem that you can't easily dismiss is the fact that we have a lot of heterogeneous data stacked on top of each other to define who you are as a biological entity. It's a little bit like the whole GPS system where you have a map that is literally two-dimensional where the roads are. You put the third dimension of hills, then you put the postal codes, the structures, the census maps, the vegetation maps, and then all of a sudden it becomes a living, breathing entity that gives you tremendous knowledge of how a city or a town, an area actually works. And so that's what we think is going to be the great challenge, that we have individuals with the genotypes, with their microbiome, with their epigenome, with their phenotypes, how they look, the metabolites that actually they produce in any particular time, over a longitudinal space. And so that dimensionality truly defines who you are, and that's actually the challenge that we have. So I want to thank you very much for this opportunity to talk to you all, and I look forward to a lively discussion in our session. So thank you.